Longevity genes, the question of control. Are there really longevity genes, switches in our DNA that decide how long we live? And if so, could we learn to control them? That question is driving a whole new field of biology. This series, Longevity Gene Control, explores the possibility that we'll be able to control our genes within the next few decades. We'll explore the mechanisms behind control, how they might work together, how big the challenge really is, and what it would take to make it safe. In this first episode, we'll set the stage, exploring what control really means, and why getting it wrong can be deadly. The feedback network of life. When biologists talk about gene control, they mean influence, the ability to dial a gene's activity up or down on purpose. Our genes already have built-in control systems. First, biology regulates itself chemically, glucose, insulin, and hormones shifting the balance of signals. Beneath that, genes respond through their own layer of control. Transcription factors and RNA fragments deciding which switches stay on or off. Together, they form a living feedback network, tuned by evolution for survival, not longevity. To shape our genetic future, we'll need to override or refine those systems, to fine-tune them toward our own goals. Layers of synthetic control. It's tempting to think we could just plug in a few new genes and call it a day. Problem solved. Eternal youth unlocked. But that was the first thing scientists tried. They found so-called longevity genes and turned them up. Doubled telomerase, boosted growth pathways, and it didn't work. Cells kept dividing, tumors formed, and real longevity never appeared. Too much is no good, too little is no good. That's why real gene control means more than adding or deleting gene packages. It means building new layers of regulation around them. So the question becomes, do we have the tools to make that new paradigm real? What tools will we explore? In this series, we're going to explore a whole new generation of tools, technologies that are redefining what biology can do. Some are already here: gene editing, rewriting the code of life letter by letter, messenger RNA, delivering temporary instructions that can teach our cells new tricks, designer drugs crafted to nudge or quiet entire genetic pathways, and transmembrane switches, tiny protein gates that respond to light or chemistry, giving us direct control over when a gene turns on or off. And that's just the beginning. More tools are coming. Molecular circuits, Yamanaka factors that can rewind cellular age, and designer proteins that help biology adapt and respond in real time. But how can we put it all together? How will we put it all together? Later in this series, we'll take on the bigger question. How can we possibly coordinate all of these tools into one system that actually works? Imagine for a moment an orchestra in a grand hall. Hundreds of musicians, each a master of their craft. They know their parts, when to enter, how loud to play, how long to hold a note. And when it begins, it's like a living machine producing something that feels like magic. They can all work together because they take their cues from the music itself, from the conductor, and from each other. Just like that orchestra, we'll have to build the tools that complete the circuit, creating a continuous feedback loop that connects the system back to itself and keeps everything in harmony. We are closer than you think. In this first episode, we've seen how evolution built its own feedback systems, and how early attempts to change them often failed. Now, new tools are giving us influence at every level of life. But coordination is the next frontier. To make it all work, we'll need a framework that turns data into control, real-time information streams that tell the system when and where to act. That's where the information systems approach comes in, the missing piece explored in Darwin's Machines 2024. It's about finding signal in the noise and using it to solve complex problems in fields like biology and longevity. Join us for part 2, where we begin exploring the first real methods of control.